So I'm really pleased that you all have made time out of your day to stay for the afternoon session to hear about this project, the TRIA Project. This is a project that got its genesis, I think, late 2006, just after the massive in-flight of mountain pine beetle into the Grand Prairie area. There was an email put out, is there anybody interested in the province in applying genomics technologies to the mountain pine beetle problem? And I sat there and I looked at that email for a good long while, knowing that there were lots of people who were quite qualified to do that. But as a recent arrival here in Alberta, I thought, well, why not? I'll put my ticket in the lottery and see what happens. And through quite an amazing concurrence of events, including the willingness of the province of Alberta and the province of BC to fund this project, the willingness of people to work together who had never worked together before, both in this province and in British Columbia, the willingness of people to work on systems that they'd never worked on before, the willingness of people to join meetings when their children were 3 weeks old and say, well, I don't even know if beetles will fly on mills, but you'll see today that they can, and they do. People willing to invest a huge amount of time to make a team project work gets us to where we are today. And there are a lot of people, as you're going to see in the next few slides, that have contributed to this project over the years. But what's really important is that this project doesn't stand alone. It has benefited enormously from consultation with ASRD. It's benefited enormously with consultation with other faculty members and other researchers right across the country. One of our most valued collaborations, I think, is with a genetics collaborator at the Canadian Forest Service in Quebec. This is a project that if I put everybody up that contributed, it would be in 2-point font, and it would be about, well into the hundreds of people. So we stand up here and tell you a little bit about this project, but what we really want to do is engender a discussion. So rather than treat this as a lecture that's coming from the podium on down, what we really want to do is open the door to questions on what you see, or comments, or any sort of discussion point. And if it takes us away from where the slides are to a new area, that's fine. If it takes us on a tangent, we'll find our way back, or not, that's fine too. The other thing is that although we can't pack all 340 people involved in this project in the audience, we have a few key people here today, particularly postdocs, that are super important to this project. And so when questions come up, those questions might be answered by somebody standing up here, or we might have a better answer coming from the people who are in the audience who have been participating, because no one part of this project has been carried out in isolation. So I really want to encourage people to speak up if they have a point, and if we spend a lot of time on that point, that's fine, that's great. So here's our outline of the afternoon. And you can see there's lots of people listed, and I've got an agenda up here that will hopefully keep us on time. I have to mention that one of our members of our project is from Newfoundland, so timekeeping is kind of an issue. If we go a little bit over, it's not a big deal. I'm not the one from Newfoundland, though. So overall, we're going to have Barry bookend us, setting the context for the project in terms of a pest management perspective, in terms of a risk perspective, and then coming back to talk about some of the research that he's involved with on environmental risk modeling, and also standing in for one of our team members who couldn't be here today, and that's Grant Howard, to talk about the economic risk assessments that link into our project. And so this will be something that you'll see as a theme through today's talks, this afternoon's talks, is that we have some fundamental biological questions, some knowledge gaps that guided what kind of research questions we designed in order to address the question, what would mountain pine beetle do in Alberta once it arrived, particularly into novel habitats? And those questions were designed specifically to fill knowledge gaps that were important for risk assessment, whether that risk assessment was environmental risk assessment or whether it was an economic risk assessment. And so by bookending, we're hopefully able to show you how these experiments that we're going to talk about and these outcomes that we have tie into that overall framework of end use, and hopefully they will tie into what you deem as germane to, particularly the operations people here, germane to what your activities are on the ground. So as Keith mentioned, this is a large project. It is an integrated project. These arrows are real arrows of connection between these areas. So the physiological genomics and the population genomics being anchors for connecting the idea of populations, using markers to look at populations, and using a different type of marker then, gene markers, to look at physiological aspects of the trees. We also looked at the beetle and fungi with other team members. We're not going to talk about that today. Tying in a whole bunch of other different experimental approaches really to feed those risk models, involving a lot of different institutions, and not neglecting the fact that this is an integrated system, so it's very difficult to study one of those components in isolation, but with the team we have the privilege of working on multiple aspects of this system, and also recognizing that there's a whole bunch of other players that we don't have here. There are mites, there are bacteria, there are yeast, that all affect how these guys interact one with another. And if we had 3 more days, we could cover them too. So this is a list of many of the key people who have participated in this particular project. And I want to just highlight the people whose names are in bold, who have done much of the work. They're the people on the ground for the project that projects you're going to see today. And what we are missing on here actually is another slide that showed the full team project of BC and Alberta, which has the 2-point font, which includes about 50 or 60 more people. So this is not a small project. Our main funder is down here, and it should be mentioned that that initial seed money that enabled us to get off the ground came from SRD and came from AFRI, and we really appreciated not only those funds to get us started, but also the guidance on how to get this project launched. So here's our laundry list of the entire team, both from BC and from Alberta, including people whose names are in gray who have moved on to other parts and other projects and other institutions. Okay, so now I'm going to turn the floor over to Barry, who's going to give us context for this project, and then I get to come back and start off the next section, which is on interactions of Mount Pine Beetle and the fungi with their pine cones.